Hello, and welcome to Shane's Microscope. I'm Shane, and this is my microscope. Let's take a peek. Today's sample is a spinach leaf. Not a decomposing one this time, though. And I really wanted to look at the cross-section of the spinach leaf, so I folded one up into my microtome and got a bunch of little slices. I'm kind of going for the shotgun approach here. I don't have any way to control which direction these are facing, so I'm hoping that if I put enough on a slide, at least one of them will be facing sideways. Spinach is known scientifically as Spinacia oleracea. Very fun to say. Spinacia is from the Latin spina for spine itself, possibly from the Persian Spinach, and oleracea is from Latin olus, vegetable. As you can see, I've got quite a collection of spinach slices in here, and I don't know yet which ones are facing the right direction. I see some with long-looking cells and some with round-looking ones, and I presume those are orthogonal to each other. So let's check one of them out first. I think I see some epidermal cells on there. Yep, there's a stoma, multiple stomata. That's from the Greek word for mouth. That's how the plant breathes. Some plants, especially many trees, have stomata only on the bottom of their leaves. Some floating water plants have them only on the top, and many plants, including spinach, have them on the top and the bottom. So that means we are looking at a piece that's facing the wrong direction. So let's check out another piece. This one has the cells oriented in the other direction, so I think this is exactly what we're looking for. Yes. So we can see that we're looking at a slice from the side. Yes, we're looking at a slice from the side. So we can see that we've got kind of this grayish layer going all the way around, some globular cells above that, and some stacks of cells above that. So this is exactly what I was hoping for. We can see some distinct regions in the leaf. Now at high magnification, we can see that one cell layer thick epidermis. That comes from the Greek epi, on top of, and derma, skin. And that's a protective layer around the entire leaf. And then above that, we've got the spongy mesophyll. Spongy comes from the Greek spongos, for sponge. And mesophyll is from the Greek mesos, middle, and phullon, leaf. And we can see that they're sort of strangely shaped to leave a lot of space in there. That's so that it's lightly packed for efficient gas exchange. As we go around, oh, there's a stoma. There's another stoma. Alright, now let's check on that top layer. We are now in the Palisade mesophyll. Palisade is from the Latin palus for stake, a series of which makes a fence. And that's why it's called that. It, kind of looks like a palisade fence. And the Palisade mesophyll goes on the top of the leaf, and the cells are arranged that way to be very tightly packed so they can get the most light to the most chloroplasts. And chloroplasts are those little green dots. That's from the Greek chloros for green and plastos for formed. And that's where the plant does its sunlight processing. I think it's interesting that they form those little clusters in there. Well, I thought it was really cool to be able to look at a cross-section of the spinach leaf. I wasn't entirely sure how well that would work out, but I think it's always worth experimenting because sometimes you get exactly what you're looking for, which we did. Alright, well I think we'll leave it there for the day. Thanks for having a look with me. Until next time, keep on peeking.